lot for the invitation. So my talk, as was already said, is focusing on data-driven molecular design in computational toxicology. And the work I'm presenting today is really work I've been doing in the framework of my role as a group leader at the University of Vienna, and not so much about what I'm doing at EBI. So in my group, we basically try to answer research questions related to toxicology mostly. So also, on the other hand, we do large-scale data analysis studies that look on trends in literature data, for instance, but the other part is mainly focusing on toxicology-related questions. And this is what I want to report today. So what are we doing here? So we use computational chemistry methods really to answer those questions. And then depending on which research question at hand, we use different methods. And basically all the time they include methods, different methods of cheminformatics and data science. So really looking into the data, into the ligand data, collecting first all the data from the available literature, and really already trying to extract some trends in the data here. But if we want to also, or if we really have also a specific target that we are focusing on, or a group of targets, then we would also consider structure-based molecular design approaches. And actually in the last few years, we have been really trying to combine those two approaches. And this was quite successful, as I will show you on the next slides. So what we are actually doing are two types of approaches. And I want to bring you each an example for each of the approaches. So the first one is really focusing on more target-centric approaches. So what we are doing here is, for instance, looking at hepatic organic anion transporting polypeptides. So that's the research question at hand that I want to show you, where we really applied ligand and both structure-based modeling for elucidating ligand binding and selectivity. But then the second approach that we are also focusing on is more a target-agnostic approach. So imagine there is a scenario where you want to model a toxicity endpoint. And in this case, the endpoint I will show you is about hepatic steatosis. And you have, for instance, an in vivo data set, but you don't really know what are the targets involved here. So also then you can do some modeling, you can build some predictive models. And so these are the two main approaches where I want to give you some examples for. So let me first talk about the first approach. So this is focusing on a project that we have been working on for the last 4 years, and this was funded by the Austrian Science Fund. So this was quite a successful project. And the main focus of the project was really to elucidate the molecular basis of ligand recognition and selectivity of these transporters. So what are hepatic organic anion transporting polypeptides? So as you can see on the left-hand side, so they are located on this basolateral membrane of hepatocytes. And interestingly, most of them, so OATP1B1 and 1B3 are closely related by sequence. So they share 80% of sequence identity. And OATP2B1 is only related by 30%. But still, they share a lot of ligands. And then there are other ligands that are really just targeting one of the three transporters. So when starting this project, we were really interested what drives this selectivity trends on a molecular basis. And why is this actually so important? Because these transporters are really important for proper liver function, but also for drug elimination. So they transport on one hand endogenous compounds like bilirubin and xenobiotics, and bile salts, but they also transport a wide variety of different drugs. And it's really interesting that they're so promiscuous, right? And it's to date really insufficiently understood how this ligand transport is working. So why is it actually important to understand this? Because if we get a knowledge about these transporters, then we can in the future prevent probably better clinically important drug-drug interactions. And one other focus of this project was once we have established structural models of these transporters, we can potentially also screen for new compounds, potentially more selective compounds even that we can use as tool compounds for future studies. So this is where we started when we started this project around 4 years ago. So we collected first the knowledge that was available in the open domain. And on the left-hand side, you can see the knowledge that my former PhD student, Alspeter Türkova, collected from literature resources. So we knew that these transporters have 12 transmembrane helices, and then we collected all the different amino acid residues, so knowledge about those residues that are involved in transport function and or really important for ligand binding. So you can already see that some of the transmembrane helices seem to have been studied a bit more heavily, like transmembrane helix 1 and 2, and then TM10 and 11 and 7. And also there have been more amino acids found to be involved in ligand transport. So this was basically the starting point. And then also from this more functional perspective, we didn't know a lot. We just knew they are sodium independent uptake transporters. They belong to the SLC family, so solute carriers. And probably a low extracellular pH would enhance OATP transport, but this exact transport mechanism wasn't really known. So the hypothesis is that they act in this alternating access mechanism. So that's called a rocker switch mechanism that you can see here. So it when the substrate binds so first it's in an outward open conformation, and then when the substrate binds, the transporter closes to an occluded state, and then is open to the inward in the end to release the substrate. And that's where we really started. There was no crystal structure available, and still there is no crystal structure from the whole OATP family. So we really had to come up with a quite fancy, so to say, pipeline for structure-based modeling. And so here on this slide, you can see the different modules and collaborations that we had during this project. But really the main focus of the project was on this pink bubble that you can see here, so structure-based modeling. And Augmented really developed a very nice systematic pipeline for doing this structure-based modeling. But we also had a very strong experimental testing partners. So this was Gergely Szakás from the Medical University of Vienna and Csilla Laska from the Hungarian Academy of Sciences. So really, of course, it's always important that when you predict something by molecular docking, for instance, that you also do experimental testing afterwards. So I would say that was the main focus, but still we had some other very integral parts in this project. And you see in the middle data mining. So that's why my talk is called data-driven molecular design. So what we always try to do in my lab when we start a research project, we first look at the available data in the public domain. We integrate data from several sources. We analyze the data, and we try to already extract some trends from this available ligand data, because this might save you a lot of time later during the structure-based modeling. And we also had some nice collaborations with other labs. So we did some proteochemometric modeling with Sharaf van Westen's group. We did a study on QSAR, a study on modeling only the APS-drawn part of this data set. And Ulf Norinder also participated and did some conformal prediction modeling together with us. So as I said, data mining is a very integral aspect. So how we started this project was really integrating data from several different open data sources, what you can see here in the left upper corner. So we built an automated workflow for integrating and analyzing the data. So data was collected from DrugBank, Campbell, Metrobase, Transportal, and EOFAR, and also from a few literature resources. Then we did compound standardization, and then we collected really the bioactivity data, and therefore got a nice data set. So we collected once a sparse data set for each transporter, and then what we call a dense data set. So what we call a sparse data set is basically all the compounds measured against any of the 3 targets, but there might be, of course, holes in this metric. So there might be some activity values and measurements missing. But then we also extracted a so-called dense data set where we would have measurements against all 3 targets. That would then really allow us to look into selectivity profiles. So then next, before we even started the structural modeling, we looked into scaffolds. So do we see a trend in scaffolds? So do we see enriched scaffolds in the data set that would allow us already to get some ideas about how selectivity might work here on a ligand basis? And then also we built some first machine learning models, and we did this in a sequentially binary classification approach, how we called it. So first establishing a model for all 3 transporters at once, so a model that can distinguish between hepatic OATP inhibition and non-inhibitors, and then 3 separate models. But really, these models weren't really built for using them for vitro screening or so later, so for screening for new compounds, but more for interpreting which features drive the separation here, and then really gather more knowledge about those different targets of interest. And one might ask, of course, this tedious data collection task, was this even worth it, right? Because you might imagine that most of the data is in Campbell anyways. And well, this might be true for inhibitors, as you can see on this slide, because most of the inhibitor data was collected from the Campbell database. But if you look into substrates, you see that Campbell is only a minor data source here for substrates. So in that case, it was like 25% of all substrates where the main source was Campbell, but then others were really mainly coming from Metrobase and Drug Bank. So this tedious data integration task really had the advantage that we could enlarge the data set, but also we could obviously enlarge the chemical space, as you can see here. So we looked into several physical properties. I'm now only depicting one property, molecular weight, but you can already see that among these 5 different data sources, it seems that a little bit the data sets differ and they have a different focus. So we also saw this for other physical properties. So it really seems we can enlarge the chemical space. And also what we gather is more confidence in our data. So because what you can see on this plot here in the right bottom corner is how many bioactivities or measurements did we find for each of the 3 transporters per compound. And you see that there were compounds in the OATP-1B1 data set that had more than 150 measurements reported. And why is this actually important? Because we later want to use these collected ligands for building classification models. So you have to set a threshold at some point and classify them into actives, inactives. And if you have, of course, more reported measurements in different assays, you can be more sure about that, about the decision you took about your threshold, for instance. So if we would see clashes here, we would then also remove this compound from the data set. So it seems that it really made sense to integrate the data. And this is also shown on this slide, actually, where you can see that this really shows scaffolds coming from other data resources than Campbell. So you see these large macrocyclic rings, for instance, that come from Metrobase. Interestingly, these are largely substrates. So we were quite astonished to see that. But it really seems in terms of chemical space, we could here increase that quite a bit. And then after collecting all the ligands, as I said, we built those first classification models. I already explained this, I think, a few slides ago. So what we did here is we built these machine learning models based on 26 physical properties and used a random tree as a base classifier. And this level 1 model, what you can see here on the right-hand side, is, as I said, is a data set of general non-inhibitors. So this would be compounds measured inactive against all 3 targets, plus the actives are basically any inhibitor on any of the 3 targets. And then we have the level 2 models, where basically we wanted to predict each and every target separately. And what we were interested in is if we see basically different features popping up here to be important for model building. So we used a feature selection method implemented in Veka. And we really saw that, okay, whereas that's not astonishing, that size, molecular weight seems to drive these trends or the predictive power of the model, but it looks a bit different for the individual models here. So for instance, we could identify molecular refractivity as an important parameter, and that hasn't been reported before. And that really actually makes sense because this is like polarizability, right? Has something to do with the charge distribution on a molecule surface. And together with the polar surface area, this seems to make sense a lot in the framework of this organic anion transporting polypeptides, where charge really seems to play a role. So we, at this stage, really before actually doing structure-based modeling, we already found out that hepatic OATP inhibitors seem to possess a higher lipophilicity, higher molecular weight, polarity, and flexibility than non-inhibitors. But then looking into planarity or like 3D-ness of the molecules, we found that OATP inhibitors are less flat and less aromatic than non-inhibitors, but it was the other way around for OATP2B1, where inhibitors are more planar. So that was an interesting trend that we observed. But I also have to say the OATP2B1 data set is much smaller than the OATP1B1 and 1B3 data sets, and might be also a little bit more focused on a certain, because we found here more planar compounds in the inhibitor data set. So we shouldn't forget about this as well. And then we came to the actual main focus of this project. So first really looking into ligand data, what trends can we extract? And then we started to build up our structural modeling pipeline. And this pipeline consists of 4 different steps. And we're quite proud that we really built this up with only open source tools and software. So we applied a fault recognition method here to identify the best template for our proteins, because as I told you, there's no crystal structure available. But in addition to this, there is no template really available with a high sequence similarity. So the closest homolog in PDB has only 15% sequence identity. So that's really a challenge. And this is also why there haven't been a lot of structural models being reported beforehand. Okay, so we have chosen the threading method. We used PGen threader here and identified the Foucault's transporter as a suitable template for all 3 transporters actually. And then in the second step, we applied normal mode analysis. So I will explain you on the next slide how exactly that works. But basically we did this because we had 2 reasons to do this. So one was we wanted to sample different conformations of this transporter, because of course these aren't static proteins, they are in motion, right? So we wanted to make sure we have the right conformation for docking. But also we wanted to see, can we identify distant relationships based on the protein dynamics? So by doing this normal mode analysis for a whole group of structurally related proteins, do we see if the same transmembrane domains move more than others, right? Because this could give us already hints about the function and dynamics of these proteins. And then the third step was the model generation step. You see, we have built up a lot of different models per transporter, also on the basis of the different conformations that we have extracted beforehand. And then we did ensemble docking, but I will explain this a bit more in detail on one of the following slides. And we applied here enrichment docking. So really seeing which model can best separate inhibitors from non-inhibitors and decoys, and therefore really we wanted to base our decision which model to choose really on these statistical tests. So the normal mode analysis, I promised to explain this a little bit more. So what this is a coarse-grained method basically. So what you do is here you present your protein by an elastic mass and spring network. So the C alpha atoms are basically the nodes, and then the springs are represented they represent the interactions between the nodes. And this is a very nice method to calculate these vibrational modes and the protein flexibility. And we have been using the software ProDye, a very nice software package that I can only recommend by Yvette Bahar's lab. So a collection of Python scripts. So really our aim was to really base this only on open source software. So now coming to our results, right? So did this actually pay off doing this large sampling study and extracting different conformations? But what you can see here is on one hand, this collective motions of protein domains. So what we did here is we looked into this protein dynamics in a whole family of 45 related major facilitator superfamily proteins. And you see the root mean square fluctuations here in blue overlaid for all the 45 proteins. And you see basically the fluctuations are the same. So it seems that always the same transmembrane helices are moving. And we have color coded them here on the left-hand side. So we actually saw the largest fluctuations in this transmembrane helix 1 and 2, which is here in this greenish color, but then also a little bit in TM7 and 11. And interestingly, if you remember what I've shown you in the beginning, these are also the domains where there have been most amino acids residues being reported to be obviously involved in binding, ligand binding and function of the protein. So that was quite interesting to see. And then looking at our OATPs, so here you can see overlaid in gray the template structure, and then in the 3 different colors, the 3 different proteins that we are looking at, and you see how much they are shifting these transmembrane helices. And you see the largest shift you see for TM1 and 2, and here you also have it in numbers for the different RMSD for the helices. And we really saw that on one hand, as I said, TM1 and 2 moved quite a lot, and then 7 and 11 little bit less, but still. But then OATP-2B1 really showed the largest deviation, which was also quite interesting to see. So this was really even before starting the molecular docking procedure, we already thought, okay, there's something going on here. Obviously, TM1 and 2 are interesting to study further. They might have an impact on ligand accessibility. They might have an impact on ligand binding. And then we performed our model generation and ensemble docking approach. As I said, we built a large number of models. So we really wanted to have, as much as we can, an unbiased approach here. So we sampled 1,000 alternate conformations of our template. So this was really, maybe I didn't mention this before. So really the conformational sampling was based on the template and not on the models that were later built. So 1,000 different conformations. And then we selected 20 different conformations on the basis of RMSD. So we selected the most diverse structures. And then we built 100 models per conformation, which led to 2,000 models per transporter. And then we looked on some quality criteria like the small property score, right, which looks at clashes, Ramachandran plot, and so on, to really sort out the low quality models. And we ended up with 60 models per transporter. And then we docked into the 60 models, our ligand data set that we have collected previously, plus decoys to have a larger set of inactives. And then we did 2 rounds of molecular docking. First, we docked into the whole protein and then selected just the 5 best models on basis of the AUC. And then we restricted a little bit the binding site and docked once more, and then really could select on the basis of really the ability of the model to separate actives from inactives. We selected really the best performing model here. And although all the models have been built on the same initial template, via going this step via conformational sampling and so on, in the end, we ended up with a different model for all the 3 transporters. And now some results from the actual work. So looking a little bit closer. So I think I didn't show you this, but when doing the scaffold analysis and looking into our data set, if scaffolds could actually drive a selectivity trend here, we found out that the steroid scaffold actually, that compounds with the steroid scaffold are usually pan inhibitors, or they have the potential to interact with all 3 transporters. And luckily, our collaborators, they had a nice in-house data set of 13 alpha epiestron, so epiestron derivatives, and you can see the structures here. So we had different variation in position R2 and R4. So here phosphorylation, halogenation, but then in position R3, instead of this hydroxyl moiety, we also sometimes had a methoxy or a benzoyl oxy moiety. And then we had, of course, our public data set that we collected, which also contains steroids. And first, we docked all of the compounds into our model, except for this in-house data set, to just in the first round see where is the binding site. And here you can see in blue, this is OATP-1B1, in pink, that's 1B3, and in green, OATP-2B1. And somehow they look like they're in the same region, but still not exactly the same. So we really clustered the poses and found out that OATP-2B1, here the binding site is a little bit more located in the N-terminal domain, so really in between transmembrane helix 1 and 2. And for the other two transporter, this binding site or cavity also exists, but in addition, there's more this central cavity in addition. And now really some very detailed results, because in this project, really, we were interested into finding reasons for selectivity. And we were really always also looking at literature data. So what do pharmacological experiments tell us? Which residues have already been reported to be involved in ligand binding? And here are two examples. So for instance, on the left-hand side, estron 3-sulfate seems to be an OATP-1B1 selective ligand. And we think we have potentially found an explanation why this is the case. So on one hand, it's binding to this tyrosine here in transmembrane helix 7, and it binds in a way that it's really buried in a hydrophobic cavity down here. And then it seems that certain hydrophobic residues are playing a role to stabilize the compound. And for instance, this isoleucine 353 here is really actually a known site of single nucleotide polymorphism. And it seems to lead to a decreased transport if it's mutated to a threonine. And then the second interesting ligand here, just as an example, in OATP-1B3, we found digoxin to be a selective binder to OATP-1B3. And you can see the reason for this behavior on the right-hand side here, because digoxin carries a large sugar moiety. And this side chain is really difficult to get accommodated. And we found that it's accommodated in a way that is only allowed by OATP-1B3 binding, because there is a glycine in position 45, which is also non-conserved and is an alanine in OATP-1B1, but a serine in OATP-2B1. So this wouldn't allow really accommodation of this side chain. And even more interesting than were our results for OATP-2B1 activity and selectivity, because this is now our in-house data set that hasn't been published beforehand. And as an example, on the left-hand side, we see a phosphonated estron derivative. And we found basically that this was binding in the way you can see here, that also serine is an important interaction partner. And then this benzoyl oxy here is again buried in a hydrophobic pocket. And we also saw that analogous compounds who don't have a benzoyl oxy here, but a hydroxy or methoxy group, that leads to decreased affinity here. And even more interesting were the halogenetic derivatives, because here we found interaction with a threonine, so a halogen bond formation. And this residue actually hasn't been reported beforehand in literature, but it's also non-conserved. So it's an alanine or serine in the other two transporters. And this compound actually is really selective towards OATP-2B1. So it's obviously inactive to the other two transporters. And we also couldn't find any docking poses for this compound in the other two transporters. But because in the beginning, I said we are combining often more the machine learning methods with structure-based approaches. So I wanted to show you a very recent example. That's a paper we have just submitted, and you can find it on ChemArchive if you are interested. So this was a nice collaboration with Ulf Norinder and Gerard van Westen's group. So what we did here actually, so collecting the data in our earlier studies for OATP-1B1, 1B3, and 2B1 ligands, we now built more fancy machine learning models than we did beforehand. So we also used proteochemometric descriptors, and we used boosting, and on the other hand, conformal prediction to account for the imbalanced nature of the data sets. So we built up 16 machine learning models, and then we used them to pre-screen our data. So we had we used a subset of the enamine library, so basically the virtual HTS collection, and then used our 16 models to predict those compounds. So we predicted so we then scored the compounds that were predicted, and we selected 250 for each of the 16 models, and then removed duplicates. So this led to 3,300 compounds that were pre-selected by the machine learning approaches. Then Alspeter docked all of them to our models, and then we ranked them on basis of the docking score. And then we did some further filtering steps. So on one hand, what we learned in the beginning already about the physical properties of the literature compounds for those 3 transporters. So we actually really filtered out compounds that were not falling into these known ranges of physical properties. We also did a substructure analysis and tried to have a bigger diversity, chemical diversity of our compounds. So in the end, we selected 44 compounds for initial screening. So that means the compounds have then been tested by our collaborators in 3 different concentrations. And after that, we manually selected 6 compounds out of those, which have then been tested in 8 different concentrations. And so this was a manual selection, as I said. So it means we really looked into potency in these initial screens at potency, but also selectivity, because in the end, we wanted to have probably new tool compounds, so compounds that have really high selectivity against one of the targets. And interestingly, as you will see here, all of these 6 compounds were active, at least against one of the 3 targets. So all are active on OATP-2B1. And the most potent compound, C7, is 40 nanomolar active. So it's has an inhibitory activity of 40 nanomolar. So that's really comparable to the most potent compound reported in literature so far. And then there are 2 that are selective for OATP-2B1 here. So this G4 and B4. And then there are 2 compounds which are pan inhibitors. So what we then further did in these papers, we looked into really closely into the binding modes and wanted to find an explanation again for selectivity. We have learned already from the previous studies a few things about these transporters, but really with respect to steroids, and now we have a larger diversity of the ligands. So looking closely at the binding sites, we found out what you can see here in this picture A, is that it seems that the aromatic residues are differently located here in the binding site. So the green structure is OATP-2B1. So you see aromats are more in the central cavity, which means the ligands are usually sitting above this, especially this residue histidine 579. So they're sitting more in this more N-terminal region. Whereas it's the other way around for OATP-1B1 and 1B3, where there are aromats more in this upper part. So ligands tend to bind below this residue here, this tyrosine 352. And this is also what we saw previously when we clustered the poses after docking all the compounds, right? That it seems that for OATP-2B1, binding site is shifted a little bit more towards the N-terminal domain. And what is also what also is an effect of this is that it seems that within or in between TM7 and 8, there is more space because there are no aromats here. So it seems that compounds can much nicer accommodate here and really form this binding pose that looks like a shape complementarity of these ligands. So if you want to know more about this, download our preprint. So there's much more detailed information, but it seems that this was really very successful, and probably that was really a combination of proteochemometric modeling, which includes protein descriptors, right? But then also combining it with virtual screening, like molecular docking in the classical sense, but also combining it what we knew already beforehand about our ligands and the proteins, right? By collecting all this information from the public domain in the very beginning of the project. And now very briefly, I want to talk about a second approach that we have been focusing on in my lab. So this is now really, I call it a target-agnostic approach. Of course, targets play a role here, but it's more if you have like an in vivo data set and you want to model an endpoint and you don't know so much about the really important targets for this toxicity endpoint, right? And before I show you my example, I just want to introduce you, because not all of you might be familiar to this concept of adverse outcome pathways, which is actually a concept that is already around since more than 10 years. It's also a concept supported by OECD. So on principle, AOPs are structured representations of biological events like molecular initiating events and key events that lead to an adverse outcome. So it might remind you of a regulatory pathway, but it's not, it's something else. So it takes another viewpoint. So the anchoring point is here the adverse outcome, and then actually each key event is examined by its importance and necessity for the adverse outcome. So it's quite a different concept, but sometimes looks pretty similar just from the perspective of representation. But what is really cool about this concept is that it's a nice framework for integrating data from different biological levels, right? So it's not targets. So it's not that all of these key events have to be targets. So it protein targets. I mean, it can be molecular key events, but it can also be cellular or organism level and so on. So you can integrate a large variety of biological data into this concept, just as a basis for understanding what we did here. So that was also a collaboration with Ulf Norind, and I've seen he's also in the audience, which is nice, I think, today. So I'm hoping he will not ask a lot of nasty questions. So the starting point was a AOP network that you can see here. So an AOP network is a condensation of different AOPs. And because in the end, it was different molecular initiating events, which you will recognize are nuclear receptors. So upon activation of these nuclear receptors via certain key events, they all lead to a common adverse outcome, which is liver steatosis. And what we had at hand was an in vivo data set, as I said, of more than 1,000 compounds, which were measured in rodent studies with repeated oral exposure, which have been collected from mostly open data sources. Some were closed as well. And you also can see, like in this depiction on the left, that this is quite an imbalanced data set. So we had a lot more negatives than positives. But still, we wanted to try, can we generate a predictive model, right? By using meta classifiers such as bagging with stratified undersampling or conformal prediction to account for the imbalance in the data set. But in addition, what we also did is we wanted to see if we include as features in our matrix also predictions from nuclear receptor models and transporter predictions, would the model improve? So I have to, I think, explain this a bit more. So knowing that nuclear receptors are playing a role here in this AOP, and colleagues from this EU Tox Risk project have actually built in silico models already for these targets, we could actually take all of these models, so these in silico models built upon in vitro data, then predict our in vivo data set, and the predictions we can use as features for our machine learning models based on in vivo data. So as additional features. And we did the same for transporters. So we knew that APC transporters like P-glycoprotein, BCRP, BSEP, and our OATPs play a role in liver toxicity. So we also included predictions from these transporter inhibition models. And interestingly, so we wanted to see what is the influence of the descriptor combinations. So will nuclear receptor predictions, transporter predictions lead to a better balanced accuracy of the models? And this is actually almost my last slide to come to an end. So it's really difficult to model such endpoints, but really because we think we're still missing some information, right? It seems to be a very complicated mechanism of action that leads to this adverse outcome. So we could achieve balanced accuracies of up to 0.7, but there weren't so many differences in the predictions with respect to taking different descriptor combinations. It didn't lead to really a lot of differences in accuracies. But of course, using only nuclear receptor predictions, for instance, led to a poor model. So in the end, using a combination of physical properties and transporter predictions led to reasonable models. And then something that is also quite well known for conformal prediction is, of course, depending on which significance level you're setting, so which error you're accepting in your model, then also more or less compounds would fall into the both category. So this you also have to take into account, that conformal prediction can guarantee that your prediction is correct, but you have to accept a certain error level. And now I'm already coming to an end. So I think I could have shown you that we could successfully employ an automated protocol for protein model generation. that includes both conformational sampling and a statistical evaluation. So this via this enrichment docking. So this seems to be a quite successful strategy. And then also combining AI-based with structure-based method for virtual screening seems to be a fruitful strategy for especially those difficult targets like transporters. And but for modeling adverse effects, like I have shown you on the last slides, we need to include as much mechanistic data as possible. I think we're not there yet. We are currently working also on data integration strategies to enrich AOP hypotheses. Potentially this would lead to more information in the future that could be integrated, but we are not there yet, I think. And the most important slide is to acknowledge all the people that have been involved and also funding. So I'm grateful for FWF funding, EU Tox Risk as well, who funded part of this work. And really very nice to work in such a vibrant field these days and have so nice collaborators. I'm really grateful for this. And I'm happy to answer any questions.